Hi everyone, I welcome you all to my video. Myself Aishwarya Patel from Nargond. Today I am starting the question banks on the nephelometry principle, instrumentation and applications. On this topic, 1 marks questions, 2 mark questions and objective types and 5 marks important questions. And in that first one mark questions and answers. What is the basic principles of nephelometry? Nephelometry is based on the measurement of light scattered by suspended particles in a solution. This type questions may be asked in exams. What is nephelometry? It is a technique used to measure scattered light by suspended particle in a solution. On what principle does nephelometry works? It works on the principle of light scattering. And the fourth one is what happens to the light when it strikes suspended particles? The light is scattered in different directions. And the fifth one is name the light sources used in nephelometry. A monochromatic light source is used. And the sixth one is what type of detectors used in nephelometry. The photodetectors is used in the nephelometry. At what angle is scattered light is usually measured? 90-degree angle to the incident light. And the eighth one is what is the function of curvature in nephelometry? It holds the sample solution. And the ninth one is give one medical applications of nephelometry. Measurement of proteins in blood serums. And the last one is where is nephelometry used in environmental science? To measure the turbidity of a water. And the next is objective type of questions and answers. And then first one is in nephelometry the intensity of a scattered light is proportional to the concentration of a particle. And the second one is nephelometry is suitable for solutions turbid. The instrumental used in nephelometry is called nephelometer. The scattering of light depends on the size of the particle. Nephelometry is a type of dash analysis. It is optical analysis. The medium required for nephelometry must contain an insoluble particle. Nephelometry is commonly used in clinical labs. And the eighth one is the unit of turbidity measured by nephelometry is NTU, nephelometric turbidity units. And the ninth one is increase in particle concentrations curves dash in scattered light. It is increases in scattered light. And the 10th one is nephelometry is closely related to the turbidimeter. And the next one is 2 marks questions like this. Define nephelometry and explain its principle. Nephelometry is an analytical technique used to measure the intensity of light scattered by the suspended particles in a solution. The principle is the intensity of scattered light is proportional to the concentration and size of the particles. And the second one is describe the instrumentation of a nephelometry. Light sources provides monochromatic light, cuvette holds the sample, detector placed at 90 degree, amplifier and readout displays result. And the third one is explain the working of nephelometry. In nephelometry, a beam of light is passed through a solution containing suspended particles. These particles scatters light. The scattered light is detected at 90-degree angle by a detector. The intensity measured is directly proportional to the particle concentration, which is used for quantitative analysis. And the fourth one is write in detail the applications of nephelometry. The applications are the clinical analysis for proton estimation in serums, immunology, antigen antibody reactions, pharmaceuticals analysis of suspensions, food industries, quantity control liquids. And the fifth one is discuss factors affecting nephelometric analysis: particle size, particle concentrations, wavelength of a light, refractive index of medium. And the sixth one is compare nephelometry and turbidimetry. Nephelometry and turbidimetry. Light measured in nephelometry, scattered in turbidimetry, transmitted. Angle is 90 degree in nephelometry and 0 degree in turbidimetry. Sensitivity is high in the nephelometry, moderate in the turbidimetry. Suitable for dilute solution for the nephelometry and the concentrated for turbidimetry. And the seventh one is explain the importance of 90-degree measurement in nephelometry. The 90-degree angle is important because it minimizes interference from transmitted light and allows accurate measurement of only scattered light, improving sensitivity and precision. And the next one is eighth one. Describe the role of detector in nephelometry. The detector captures scattered light and converts it into an electrical signal which is amplified and displayed as a measurable value. And the ninth one is write advantages and limitations of nephelometry. Advantages is high sensitivity, suitable for low concentration quick analysis. Limitation is affected by impurities, not suitable for highly concentrated solution, requires calibrations. And the 10th one is explain the turbidity and its measurement by nephelometry. Turbidity is the cloudiness of a liquid caused by a suspended particles. In nephelometry, turbidity is measured by detecting the intensity of a scattered light, and results are expressed in NTU. And the next is 5 marks questions. First one is define nephelometry, explain and its principle, instrumentation and working. Nephelometry is an analytical technique used to measure the intensity of a light scattered by the suspended particle in a solution. And the principle is when a beam of a light passes through a turbid solution, particle scatters light in all directions. The intensity of scattered light is directly proportional to the concentration and size of a particle. Measurement is usually done at 90 degree to avoid interference from transmitted light. Instrumentation is light source, cuvette, detector, amplifier and reference system. Working light passes through the sample and the particle scatters light. Detector measures scattered light. Signals is amplified. Result is displayed. And the second one is describe the instrumentation of nephelometry. Light source which samples and detectors an 90 degree and displays. Light source provides the stable monochromatic light. Cuvette holds the sample solution. Detector placed at 90 degree to detect the scattered light. Amplifier and display converts the signal into reliable forms. And the third one is explain the principle of nephelometry and discuss factors affecting it. Based on light scattering by suspended particles, the scattered light intensity depends on the particle concentration and size. Factors affecting nephelometry is particle size. Larger particles scatters more light. Baratite containing increases scattering up to limit. Refractive index differences. Presence of impurities. Fourth one is write in detail the applications, advantages and limitations of nephelometry. First one, the applications: protein estimation in serums, water turbidity measurement, pharmaceuticals analysis. And advantages are high sensitivity, suitable for low concentration samples, rapid and simple methods. Limitations is affected by impurities, requires proper calibration. Explain factors of defecting nephelometry. Particle size: larger particles matters scatters more lights. Concentration: higher concentration increases scattering. Wavelength of light: shorter wavelength scatters more. Refractive index: difference between particles and medium affects scattering. Presence of impurities may cause errors in measurements. Thank you all.